So these are the topics we will be covering today. Salivary gland, as you know, it's an exocrine gland. It's not an endocrine gland. This gland, it secretes something, and this secretion comes through vessels. Okay, tubular structures, means ducts. These glands are actually accessory organs, glands for digestive system. We have GI tract. To support digestion, there are some glands. One of the gland is digestive gland. Actually, there are three pairs of salivary gland. Three pairs of salivary gland. What are these? Parotid gland, submandibular gland, and sublingual. Okay. Largest one is parotid gland. It doesn't mean it will secrete more saliva. Actually, it secretes only 25% of total saliva. And this one actually secretes maximum part, 75%. And some sublingual is only 5%. The composition varies. In some gland, we will find only watery content will be there, and within this there will be various ions and enzymes. These sort of secretions are called serous secretions, and from these are mixed glands. They will secrete zymogen, mucin, as well as different ions. We will compare these values when we will be taking food, and these foods will face enzymes for digestion. So it is the first digestive juice that food will face. Here in the buccal cavity, food will be masticated, and at that time, before that time, saliva will be secreted during mastication. Also, saliva will be secreted. We will come. What are the different stimulations for salivary secretions? So we have seen there are three major salivary glands, three pairs of parotid glands, submandibular, also called submaxillary, and sublingual. So these are the location. This is what? Parotid, this is sublingual, and this is submandibular. Parotid gland, it weighs around 20 to 30 gram, whereas it is only 8 to 10 gram, and this one is sublingual is only 2 to 3 gram. And their location I already told, and these glands drain their secretion through a duct. This is called Stenson's duct. Here it is called Wharton's duct, and it is Rivenna's duct. Okay, and when you take a histological section of parotid gland, you will find the cells are mostly serous, and here you will find serous as well as mucus. Mucus cells will secrete mucinogen, which will be later converted into mucin, which is very slippery, and it will help passage. This mucin will help passage of food bolus into the next part, and moreover, it will be very easy for mastication. Okay, so that is the purpose of mucin. And percentage I told, these glands are connected with nervous system, autonomic nervous system. They control secretion of the salivary glands. And these are under the control of sympathetic as well as parasympathetic. Sympathetic nerves, parasympathetic nerves that supply these glands are 9th cranial nerve, that is glossopharyngeal nerve, and 8th cranial nerve, that is facial nerve. Facial nerve. So these two nerves this is pons, medulla, spinal cord. Here is salivary nucleus. This is superior nucleus and this is the inferior nucleus. What is nucleus? The cell. Within the cell there is a controlling center, that is nucleus. Is it that nucleus? In nervous system, nucleus has got different meaning. What is this? We know nervous system is made up of nerve cells. Nerve cells are having cell body, axons, dendrites. The cell bodies, when a group of cell bodies will be staying together and will be performing a particular instruction, then they are called nucleus. Actually, this nucleus are nothing but accumulation of group of cell bodies of some neurons. From these cell bodies, axons will be coming. So here, the superior salivary nucleus, superior salivary nucleus, and since it is below, it will be inferior salivary nucleus. So they are cell bodies. So from these cell bodies, axon will come and leave medulla through cranial nerves. This is for inferior, this is glossopharyngeal nerve, 9th cranial nerve. So this is nothing but 9th cranial nerve. 9th cranial nerve. Can anybody tell the difference between sympathetic nerve and parasympathetic nerve? This is one of the common questions, no? Difference between sympathetic and parasympathetic, functionally, structurally, neurotransmitter function. So there are difference between sympathetic and parasympathetic. Both are under autonomic nervous system. Sympathetic nervous system, they are catabolic in action. Means when these nerves are stimulated, there will be functional change, repeat functions, say for heartbeat, breathing. This will, from sympathetic nerve endings, mainly epinephrine or noradrenaline, these are secreted. We know when these are secreted, when we become afraid, our heart starts beating, not at increasing rate. What is the cause? Because of stimulation of the sympathetic nervous system. From the nerve endings, there are nerve, sympathetic nerve, innervation in SA node. So it will cause release of, from the nerve ending, release of epinephrine or noradrenaline. It will stimulate SA node. And the SA node, instead of 70 or 80 beats per minute impulse generation, it will increase. It will increase the impulse generation. So in that case, it would be 120, depending on how much you were afraid. So depending on that, the heartbeat will be increased. Means at SA node, from SA node, electrical generation will be increased. Not only this, conductivity will be increased, excitability will be increased, contractility will be increased. So many changes take place under the action of this hormone, neurotransmitter. So as a whole, there will be increased activity of the heart. So if the activity of heart is increased, so there will be need of energy. So where from this energy will come? Breakdown of food, glucose. So this is a catabolic process. When but see the action of acetylcholine. See the action of acetylcholine on heart. When the vagus nerve is stimulated, heartbeat is lowered. There is slow beating of heart. Why? From its nerve ending, acetylcholine is secreted. This acetylcholine has inhibitory effect on SA node, on conductivity, on contractility, on excitability. As a whole, the activity of the heart will be decreased, activity of the muscle will be decreased. So, there will be less catabolism. So there it will be, so that's why we call, since there is less catabolism, so we call it anabolic. Okay. This is one difference. But it is not always true that sympathetic nerve ending, sympathetic stimulation will increase all the functions. Sometimes it decreases some functions. Opposite is true for parasympathetic. Sometimes you will find parasympathetic activity stimulates the function of certain glands. But in other organs, it has got opposite effect. Okay, so this is one functional difference. Second difference is the nerves. The nerves, they will be having two parts. There will be one ganglia outside the spinal cord, near the, either it may be very close to the spinal cord, this is spinal cord. So there are chains of ganglia. These are sympathetic ganglia. These are also, ganglia means there are a lot of cell bodies are there. Of different neurons, cell bodies are there. So these are ganglia, and this chain is sympathetic ganglia. If I take a cross section of spinal cord, it will look like this: gray matter, and these are white matter. And there are some cells. These cells are called lateral horn cells. LHC, lateral horn cells. Similar cells are also there here. Anterior horn cells, posterior horn cells. From brain, autonomic nerve centers, motor nerves are going. Not motor nerves means efferent nerves. So from the brain, efferent nerves will be coming down through spinal cord. It is true for sympathetic nervous system. through spinal cord it will be coming down, and from the sympathetic, from the spinal cord, they will be traveling to, in our case, for salivary gland, they will be coming through lateral horn cells. In the lateral horn cells, suppose now they are to exit the spinal cord, then they will follow the anterior route. There are two routes, from the back door and these are the front door. Okay, so these are back doors through which from periphery nerve enters, and this is the exit route through which nerves come out of spinal cord. After coming out, they will end in this ganglia. This is cell body of a neuron, and this is the axon. This means axon terminal. So this one from here through anterior door, this nerve will be coming, ending in the ganglia. From here, post-ganglionic, this is the ganglia, post-ganglionic neuron will arise and will go to the target organ. So there are two, we can see, there are two neurons. One is preganglionic, before the ganglia, and another is after the ganglia, post-ganglionic. In case of sympathetic neuron, the sympathetic nervous system, sympathetic, preganglionic fibers are short, post-ganglionic fibers are long. They are to travel since ganglia is near the spinal cord. That's why length is less. But post-ganglionic fiber, they are arising from this ganglia and going to a distance place where the target organ is. That's why its length is more. We'll find, so suppose this is the sublingual salivary gland, and it is, they will supply from this, they will supply this there. So when they will be stimulated, they will make change in this. Actually here, as I said, what do you think? Stimulation of sympathetic nerve will cause increased activity of the salivary gland or decreased activity? In heart, it increases the activity, sympathetic nervous system. Here it is the opposite. Means it will decrease salivary secretion. How do you know? When you become afraid or you are waiting outside for vibration examination, so you are very much tensed. Your mouth dries. Isn't it? When you become very afraid, your mouth dries, you want water. Why? There is less secretion, adrenaline secretion, epinephrine secretion. This inhibits the salivary gland. As a result, less secretion is made. So your mouth becomes dry. This is one good example. Okay? But in case of parasympathetic nerve, they actually arise from here through cranial nerves, they come out. And here the presynaptic membrane is long, but postsynaptic postsynaptic neurons are short. Why short? Because here ganglia lies close to the organ. So it is coming down from cranium, coming to this gland. Okay, in between there should be a ganglia, and this ganglia is very close to the, very near to the or within the gland. So first they will open into the, they will make, their axon terminal will be ending there, and from there postsynaptic neuron will be originating, and they will supply the gland. That's why its length is short. Postsynaptic neuron, presynaptic neuron. Okay. When parasympathetic nervous system is stimulated, means facial nerve, cranial nerve or glossopharyngeal nerve, what will happen? There will be increased secretion of saliva. This way, it is secretion of salivary secretion is controlled by autonomic nervous system. You, get up, come here. I think you are following my class or not? Okay, now tell me, if sympathetic nerve is stimulated, what will happen to salivary secretion? Whether it will be increased or decreased? And others will, if he answers correct, others will say, okay, yes sir. Sympathetic stimulation, decrease. Is it correct? Good. Very good. Now everybody came to know, sympathetic stimulation is, it will cause decrease in salivary secretion. And parasympathetic stimulation?
Increase in salivary gland.
Okay, good. She said there will be increase. Is she correct? Okay, good. So this way we can control, nervous system controls the secretion. So one part we understood. Another thing, secretion is controlled by some other factors. Oh, think about imli. Everybody, girls especially, imli. See what is happening in your tongue. Saliva coming out, no? So you just heard the name of imli, immediately salivary secretion started. So thinking about some food, tandoori, chicken tandoori. Okay, dosa. Rava dosa. Smells good. So immediately, by seeing visual stimulus, thinking, discussing about food, smell, all this, and all this will stimulate. Food is not there. Food is not there inside the mouth. Or it is not there in front of you. You can't see this. Even then, you have started secreting. So these are the stimulant. And while you will be taking the food, so this food will come in contact with taste bud. Oh, taste good. Yeah, I can take new food, no? Let's see. How is this? It's good. Means tasty. Means there will be immediately taste bud will cause more secretion of saliva. Okay, while you will be masticating, churning food inside your mouth, then only it will cause yawning. That will cause yawning, isn't it? Yes. She said yes. What it will cause? When you will be you will be churning the food, masticating the food, whether salivary secretion will be increased or decreased? Increased, not yawning. Yawning will not be increased. Yawning will go. Delicious food, no? So it will go. Okay, so this is the control. These are the controlling mechanisms. Chloride, bicarbonate, mainly bicarbonate. This will make the secretion basic. Okay, chloride ion will be there. Phosphate will be there. Okay. How much U? Yes, U. What is the water content of the secretion? Don't look at the slide. Look at me. 90%, 92%, 99.5%. Only 0.5% are solids. And out of this, mostly are organic. Enzymes. So, there are some lipase enzymes, no? This lipase enzyme has a very, very little function. It is negligible. Very negligible amount of fat can be digested by lipase, this lipase. The amount is very less and its scope of digesting triglycerides is very less. So we can ignore it. So we can ignore it. Okay. So what are the main constituents actually? Salivary amylase. Now before starting with the function of salivary amylase, its function is digestion of carbohydrate, particularly starch. What is starch? I am coming to biochemistry. And I know we are the very first class, it was biochemistry was your first class, chemistry of carbohydrates. So what are starch? What type of carbohydrate it is? Carbohydrates are classified into three. One, monosaccharides. Then oligosaccharides. Oligo means in this there will be 6 monosaccharides, 10 monosaccharides maximum. Oligosaccharides. And if more than 10 glucose are present, these are termed as polysaccharides. Example of polysaccharide is starch. Example of polysaccharide is starch. And these are synthesized by human body, no? He said yes. So you nodded your head like this. So who makes it?
Cow digests it.
Cow.
Cow digests it, sir.
Who makes this starch molecule? Who makes? Plants. These are made by the plants. And they can provide a lot of energy. So that's why we take it. Rice, uncooked rice, they also contain starch. Do you eat wheat? Do you eat uncooked wheat? Uncooked rice? Why not? Why not? You can eat.
So you can't digest it.
You
Can't digest it.
Why it cannot be digested?
You don't have such bacteria to digest it.
Read the first line. I will ask this question later. Why we can't digest starch that directly we get from them? Rice, pre-cooked, uncooked. What is there? What is the problem? There is a coating. And this because of this coating, salivary amylase cannot act on that. So this coating has to be removed. How it can be? When we cook, some water, it will absorb some water, so it will burst. The outer covering will burst. Now amylase will be able to enter within the molecule. You have seen the banyan tree? Everybody have seen? Hello? What do you call it? What do you call it in Hindi? Yeah? Okay. In Bengali? Bodgas. Now you think the starts as a banyan tree. So what is their stem? Having lot of branches, and on each branches there are leaves, lot of leaves, and these leaves are actually glucose. These are glucose. So glucose leaves, these branches having lot of glucose, these branches having lot of glucose, and both are linked together by a stem. Yes, madam. No. Okay. So now salivary amylase will enter within the molecule and cut not the branch. It cannot cut the branch. It is unable to cut the branch. Why it is unable to cut the branch? Because these branches are made of 1,6 glycosidic linkages. Three types of linkages can be there connecting two monosaccharide molecules. One may be 1,2 glycosidic bond. It happens in sucrose. First carbon of glucose and second carbon of fructose. They will make a bond, 1,2 glycosidic bond. Don't you know that? How come? And another bond which is most abundant, that is 1,4 glycosidic bond. It means there will be a bond connecting first carbon of the first glucose and fourth carbon of the second glucose. So 1,4 glycosidic link. It is most abundant, most common. In disaccharide, in trisaccharide, in lactose, in maltose, you will get 1,4 glycosidic linkages. Okay? Now, glucose, linked by 1,4 glycosidic linkage. 1,4. Now it is, how many glucose it is having? 3, 6, 7, 8, 9, 10, 11. More than 10. Okay. This type of, I will call them amylose, not amylase, amylose. All the glucose molecules are attached by 1,4 glycosidic bond. Okay? And one important thing for salivary amylase is it cannot break, though it is alpha amylase, salivary amylase, it is capable of hydrolyzing one alpha 1,4 glycosidic linkage. It is capable of this. This is 1,4. It cannot. Amylase cannot break this. Because this is terminal one. It can act only on central. So here it can break this. It can break this. If it breaks this, what will happen? We will get a triose. Triose. Glucose, glucose, glucose. Amylase can't do it. It can make this only. There is another compound called maltotriose. Triose, but maltotriose. Problem is what? There is 1,4 glycosidic linkage and another is 1,6 glycosidic linkage. But our amylase is helpless. It cannot break this 1,6 glycosidic linkage. That's why it cannot further digest this compound. Okay, terminal wall not possible, 1,6 not possible, so it will be left alone. This will be taken care of by the other enzymes. Okay, so this sort of polysaccharides is called amylose. Now, if it forms this compound, see this is 1,4. This is 1 and this is 4th carbon made glycosidic ligand. But first carbon is free. Its first carbon is free. With this first carbon, it can attach another glucose, first carbon and 6th carbon of this one. So this is the first carbon and this is the 6th carbon. So there will be formation of 1,6 glycosidic linkage. With this, actually what is a new branch is formed. From this again 1,4 glycage, this will be formed. Another chain. Now we'll see another enzyme is the deep branching enzyme. So this deep branching enzyme will break this. As a result, we will get amylose. Now this amylose will be broken by the amylase enzyme. If in a polysaccharide there are a lot of branches, we call them amylopectin. Amylose and amylopectin. In amylopectin there are 3,000 to 6,000 glucose molecules. In amylopectin part, where there is lot of branching, but in amylose part only 60 to 600 glucose residues. There can be 60 to 600 glucose residues. Okay. Starch molecule, a homoglycans. What is homoglycan? Don't know? On hydrolysis, only one kind of monosaccharides will be formed. Starch is a homoglycan. If you hydrolyze it, only glucose you will find. You will not find other monosaccharides. You will not find fructose. You will not find galactose. You will not find ribose. You will not find deoxyribose. So only thing you will get one type of monosaccharides. So this type of compounds are called, carbohydrates are called homoglycans. Heteroglycans means different kinds of monosaccharides you will get. Fructan means if like inulin, if you hydrolyze it, only fructose you will get. Glycan, starch is a glycan. Glycogen is glycan. On hydrolysis, only one type of monosaccharides you will get. So this is in starch. What you will get? Two things. One part is, most part is amylopectin, where we will find lot of branches are there. 1,6 glycosidic bond is there. And another part, there is no 1,6 glycosidic linkage, only 1,4 glycosidic linkage. Our salivary amylase will act on only amylose part of starch. It will enter within the molecule and you will find it will, as I said, not the terminal 1,4-glycosides they can break, only the central one. Where is Samir? Chadar? He is absent today? Sorry. One more thing. Why it is needed? Boiling, why needed? And more important thing, glucose, it has several it may remain in solution in different forms. In straight chain, we see HO, C, H2A, CH2A, CH2A, CH2A. So it is a straight chain. But it may form a cyclical chain, a structure. What is this called? Who is this person? I heard the name. Pyranose. Here it is called pyranose. 5 carbon will make a ring-like structure. Pyranose. Fructopyranose. Glucopyranose. So, in aqueous medium, they can remain in 3 forms. Either as pyranose or as furanose. In case of why it is furanose, 4 carbon will be associated for formation of this ring. If it is 4 carbon is used, then it is furanose, and 5 carbon is pyranose. And if it remains in same not a cyclical structure, so glucose can remain in cyclical structure. Okay. Now see. This is what? Glucopyranose. Glucofuranose. 1, 2, 3, 4 carbon. Here it is 1 carbon. Here it is 1 carbon. Here there is 1 carbon. So 4, 5, 6. 5. Hello? You didn't ask me? No, you're not allowed. Go there. Okay? So now see how maltose is formed. Glucose. First carbon, sorry. This is the first carbon, this is the fourth carbon. 1, 2, 3, 4, 5, 6. So there is a link. 1,4-glycosidic linkage. Now see, glucose. Okay? This is the bond. 1, this is the fourth carbon. 4th, 5th, 6th. First, second, third, fourth. And this is the first. So with this one bond is formed, 1,4-glycosidic bond. So this is one glucose. So now they wanted to make a branch. See, this fourth carbon is used, but first carbon is not used in bond formation. So now it will form a bond with the 6th carbon of another glucose residue of another. So now they will form a branch. Now again, new chain will be formed. So actually, in this way, starch molecule are formed with several branches, and these branches are made by 1,6-glycoside. You will retain in your mind these points? Okay. Now see how our amylase acts on starch molecule. Glucose. Amylase, alpha amylase acting, breaking these, but unable to break this. Okay? Here also, breaking these, but linkage 1,4-glycosidic, close to this it cannot break. The glucose residue, one glucose, they are one side they are making 1,4 and the other side they are making 1,6. So it cannot touch this 1,4-glycosidic link. This is the stages of digestion of starch by amylase. You won't get it in other book. Starch, it is maltose. Maltose means 2 glucose units. Maltotriose, 3 glucose units linked together. And erythrodextrin, more than 10 glucose residues. Why it is erythro? If you add iodine solution, it will produce red color. We know starch gives blue color. But dextrin will be confirming that yes, this has been formed when we will see the red color on addition of iodine. Is that clear to you? Okay. Then what will erythrodextrin will be further digested by the amylase into isomaltase, maltose, maltotriose and acrodextrin. Acrodextrin means no color. If you add iodine, means it is more smaller. Gradually it's becoming smaller in size. Molecule is gradually becoming smaller in size. So after the action on acrodextrin, alpha limit dextrin, here glucose is less than 10, but there are a few 1,6-glycosidic linkage. This will, our amylase will say, hands up, unable. I am unable to digest this. So other compound, isomaltose, as I said, there are isomaltose, there are 3. One is 1,6 bond, another is 1,4-glycosidic bond. Maltotriose, there are two bonds in maltotriose. For binding 3 glucose, we will need 2 bonds. So both the bonds are 1,4-glycoside. But for this, there are separate enzyme. There maltase. There is separate enzyme maltase. It will digest this. Is that easy for you? No. So what is it? So already discussed. And other functions of salivary juice. What are the functions? Mucin. It makes slippery. The food makes it slippery, easy to masticate, easy to swallow. Okay? And you will be unable to speak if there is no secretion of saliva. Your mouth will be dry. When you went out, you didn't ask me. I saw, but you didn't take my permission. How many times I have to tell you? Lactoferrin, last one. Lactoferrin, the bacterial cell contains iron. Lactoferrin removes this iron. If it is removed, the bacteria dies. So the saliva contains lactoferrin. So with this, it can kill bacteria. Okay? And there is proline, one amino acid proline. And if any protein contains proline, content of proline is much more, then it has some protective function. How? It protects the enamel of the teeth. Enamel of the teeth. And it limits the toxicity of toxin. It prevents the toxicity of toxins. So that's why it is very important. Saliva keeps our mouth moist, acid. Because of the presence of this, we can taste the food. Because food will dissolve on this salivary water content, and this soluble part of the food will stimulate the salivary the taste buds. Then we will be able to perceive how the food is. What is whether it is tasty or it is bitter in taste or it is acidic. So you can, or it is hot or it is cold. So you can perceive the food when it is, there will be salivary secretions. And IgA, immunoglobulin A, it also produce, when there is any bacteria antigen, it will produce inflammatory reaction, thereby it will destroy it. And there are lysozyme. With this lysozyme, it can kill bacteria. So there are protein. That's why the dog, when it is injured, it applies its saliva. With this, it cures its injury. Lot of buffer is there. When we vomit, when we feel vomiting tendency, very important. When we feel immediately what by increased salivary secretion is there. Before vomiting, just when we feel nauseated, that time only salivary secretion starts heavily. And when this acid will come through food or any content of gastric thing, through vomiting, it will contain lot of acid. It may destroy your epithelium. So immediately the salivary is having buffer bicarbonate ions, so it will buffer the hydrochloric acid. So HCl will be unable to damage the inner wall. Saliva excretes certain heavy metals like mercury. In different foods, through foods, we are ingesting mercury, lead. So these are harmful for the body. So the body excretes through saliva. Thiocyanates, example, these are excreted through saliva. Proline is already told. So this is about, now if you get a section of salivary gland, whatever it is, either this or this or that, you will find some tubular structures and ring-like structures. These are actually tubules, ducts. And at the end of each duct, this is one duct, at the end of it, there is a bulbular structure. We call it acinus. Acinus rather. Acinus. Acinus is singular and acini is plural. So lot of acini will find, and these are lined by epithelium. We call it acinar cell. They secrete enzymes, mucins, sodium chloride, sodium ion, chloride ion. So when these are produced here, they will be passing through the duct, intercalated duct. So while passing through the intercalated duct, it will reabsorb sodium and chloride, and in return it will add potassium, bicarbonate and chloride ion. It will not allow too much excretion of too much of sodium. So it will try to retain. This will be absorbed while passing from the lumen of the echinus will be traveling to the duct. So this will be absorbed, and in return it will produce, it will secrete sodium, potassium ion and chloride ion, bicarbonate ion. See, this is it is in your book. I think this is in AKG. Okay. Okay. I already told regarding control. I have told this is the mechanism. So it's over. Okay. And in the coming class, What is the next topic given? Tell me.